Viruses behave like living things in that they can reproduce or multiply and change or mutate. But they do these things only within specific living cells, where they're able to make use of the energy and the building blocks within these cells. And viruses are, of course, very tiny. Tens of thousands of polioviruses arranged in the form of a crystal look like this when examined by means of the electron microscope, magnified about 1,000,000 times. A single one of those particles arranged in a model looks like this. A bacterial virus looks like this. And a plant virus, tobacco mosaic virus in model form, looks like this. Virologists study viruses from many points of view. I, for example, am interested in how viruses multiply and mutate and in their relationships to cancer. Other virologists may be interested in how viruses are built or how they look when they are released from the cell. It is this latter point of view that we are going to be interested in today. It has been the special interest of my colleague, Robley Williams, a one-time astronomer who for the last 15 years has been interested in how viruses are built and has studied them from the standpoint of giant molecules.
When I consider viruses, I like to think of them as very small particles or very large molecules. What I'm interested in is their weights, in their shapes, in their sizes, in their detailed structural architecture. There's a good reason for this kind of curiosity, because like all virologists, I'm really a biologist. What I'm interested in is how do cells manufacture their stuff? How do cells duplicate? In other words, what is the living process? And the study of viruses can give us some answers to this process. A more direct reason for my curiosity is that knowledge of the detailed structure of viruses places limits on our speculations as to how they're built. Suppose, for example, that we have a hypothesis that results in a virus being built in this way, and as a matter of fact, we find out that the virus is built in that way, then certainly our speculation is wrong. Just as we can infer how a automobile factory works by looking at the details of a finished automobile, so we can infer something as to how a cell works by looking at the detailed structure of the virus particles turned out. But we can't examine virus particles from this point of view unless we have them pure. So we're going to consider how we purify viruses, in this case, a tobacco mosaic virus, which might be infected into a tobacco plant like that. And we have to go to the greenhouse. And if we go to the greenhouse, we find that we can take tobacco mosaic virus with a little carborundum and rub the material on the leaves of small plants. And we rub each leaf, and then we wait about 2 weeks, and we find that the plants have grown and that the plants have now become infected and mottled. Then we're ready to harvest the plants by just clipping them off and putting the leaves and the stems inside a bag. Now we're going to take that bag and freeze the material in it, because the freezing ruptures the cells and makes it easier to get the virus particles out of the broken cell. Then, after the material is frozen, we are ready to start to grind it up. So the leaves are put into a meat grinder, which really makes hamburger out of the tobacco leaves containing the viruses. And after we have hamburgered up a pound or so of this material, we are then ready to extract the juice. We do this by putting this stuff inside of a spinning centrifuge with a cloth filter. And we whirl the juice out of the leaf debris, leaving the large leaf material behind and having only the juice containing the virus particles and the plant pigments in a colored fluid. We now want to spin the virus particles out of this fluid, separate them from the water and from the salts and the plant pigments, and we do this by using a high-speed centrifuge. We pour the juice into little tubes, and these tubes are then put into the centrifuge rotor. We're going to spin the centrifuge at about 40,000 revolutions per minute. The rotor goes into the centrifuge, and in the course of about an hour, we'll find that all the virus particles have been flung to the outside of the tube. We're then able to pour off the juice, and after we have poured off the juice, we find that there is left in the bottom of the tube a pellet. And this pellet contains viruses in quite purified form. By repeating this process 1 or 2 times, we're able to arrive at material which is really quite pure. For example, in this electron micrograph of tobacco mosaic virus, we see that we have virus particles and practically nothing else. So it's pretty evident that we can get a solution of purified viruses. Now that we have such a solution, what we'd like to do is to measure the weights and the shapes and sizes of these virus particles. In order to get the weights, we have to use what's called an analytical ultracentrifuge. A centrifuge separates large particles from small particles, and here I have a mixture of some very small particles and some carborundum that are relatively large. And when I shake it up and then allow it to settle, the large particles, as you see, go down to the bottom. Now I can speed this up by whirling it around, which is what a centrifuge does. And you see, with just 2 whirls, the large particles have now gone to the bottom. We do this a little bit more precisely in the case of a centrifuge. Of course, we fill a small container that has glass in top and bottom with a virus solution and put this in what's called a rotor that goes in the centrifuge, and this spins rapidly, maybe 60,000 revolutions per minute. And we measure how fast the virus particles are flung down to the outside of this container. And in that way, we get an idea of the weights of these particles. But another element that has to be concerned is shape and sizes. In other words, we would like to know not only the weight of the particles, but how they are shaped. And to do this, we have to measure what we call the viscosity of a virus solution. Now, viscosity has to do with how fast a liquid essentially will pour. And I have a couple here. One is just water. The other one is water with some very large molecules in it. And the presence of the large molecules means that it pours much more slowly, the one on my right, than the plain water will pour. Now, if we can measure quite precisely this pouring rate, we have an idea of the viscosity of a virus solution, and that gives us very good ideas of the shapes and sizes of the virus particles. We can also take a straight look at these particles. And to do this, we use the electron microscope. The electron microscope uses electrons instead of light, and instead of using glass lenses for photographing light, it uses magnets in order to focus the electrons. It is rather a large instrument, and it is necessary to have a vacuum in the instrument while the electrons go through. We have first of all to prepare the specimen, and this is done by taking some very tiny copper grids which are filmed with collodion, and then we take the virus solution and we spray onto this grid the suspension of viruses, let's say tobacco mosaic virus. Now we take the tiny grid with the virus droplets on it, which have now been dried, and we bring this over to a vacuum machine which has in it a filament of wire which can be heated quite hot, which causes a film of metal to condense on the far side of all the small particles like viruses that happen to be sticking up. We call this shadowing, and these shadows appear dark in the electron micrographs. It's possible to do a job of shadowing, actually, by using a paint can, and if we do this, we find that what happens, suppose, for example, we have a tobacco mosaic virus and a polio virus, and now we can just plain shadow these things. And on the far side, or the shadowed side of each particle, we find that the paint isn't there, and so we have made a shadow. Now, if we go back to the filament and turn it off, and then we're ready to take the specimens out, we can then put them in a little holder and insert this holder in the electron microscope. Remember now, the electron microscope can magnify as much as 300,000 times, so we have to be quite precise in the handling of the instrument. We then close up the door of the instrument, pull a vacuum, then turn on the juice, and we're ready to take a look at whatever we have there. Now we can actually see what these virus particles look like. We can project the image on a fluorescent screen, such as the front of a television tube. And here we have some particles, actually, of an insect virus, the Tipula virus, and we can focus the picture back and forth and see the particles quite clearly, and we, of course, can also photograph the particles. Well, there are many ways of determining the shapes and sizes of virus particles and their weights. Unfortunately, they all agree pretty well. And the upshot of it is that we have quite a variety. For example, we have virus particles that are quite regular in their outline. We also have them as rather large spheres. We have them essentially as tadpole shapes. We have tiny spheres, and we have rod-shaped viruses. Now, if we actually want to take a look at what the viruses look like, so to speak, in real life, we insert them in the electron microscope, and when we do this, using poliovirus, we find that there is an exceptional degree of uniformity of size and shape of these viruses such that they actually crystallize. Now, we can also try another virus. For example, let's take a look at tobacco mosaic virus. Here we have the typical rod-shaped forms, but once again, a high degree of uniformity of shapes and sizes. An amusing variety is this insect virus, the tipula virus, whose outlines are perfectly hexagonal, corresponding to the polyhedral shape that we saw in the model. And last, we can take a look at the tadpole-shaped viruses, or the bacterial viruses, one of which is frozen dried, and the other two of which are air dried. So we can get a pretty good idea of the shapes and sizes of viruses. But this isn't enough. What we want to do is to find out how they are built in much more detail. Now, in 1952 and onwards, we've been able to do quite a bit about this. And one way to go at it is by electron microscopy. It's possible to take tobacco mosaic virus and degrade it gently, and then one sees that the nucleic acid of the virus has come out in the form of a very fine strand. It's even possible to look at the protein of the virus after it's been degraded, and when we do this, we see that the protein fragments have pretty much the shape of little donuts. All we have seen, however, so far is that the protein and the nucleic acid of viruses is separable, and we have seen something about what they look like. But what we want to do now is to find out how they're packed together in the virus particles. In other words, what is the fitting arrangement? To do this, we have to use X-ray crystallography. If we take a crystal of a virus and shine X-rays through it, we get a geometrical pattern where the X-rays have been scattered, and they are scattered according to the shapes of the virus particles within the crystal. So we have now quite a bit of information, both by electron microscopy and by X-ray analysis. And what do we find? What we find in the case of the spherical viruses, this is a polio model, magnified 65 million times, that the nucleic acid is on the inside of the virus particle, but most importantly, around the outside are the protein subunits, as we see here with balls, and these are arranged in perfectly regular order. There are exactly 60 in this virus, not 59 or 61. They are arranged, as you can see, in groups which create for us perfect pentagons, or they are arranged in groups which create equilateral triangles. And similarly, it is with tobacco mosaic virus, which is a rod-shaped virus, but we find when we come to look at tobacco mosaic virus in greater detail with X-rays that the protein subunits are arranged as a sort of spiral staircase. In other words, each protein subunit is like a step on a spiral staircase, and around the same spiral goes the nucleic acid on the inside. But now, why is this so? We see that virus particles are not made at random. As a matter of fact, they're made with a perfect blueprint. They are made, indeed, with a master plan within the cell. I wish I knew the answer. I wish I could really say, why do cells make viruses this way? But I can't say really why. But perhaps we can get a glimpse of why by examining the details of the protein structure and of the nucleic acid structure. But we've wandered a long ways from reality, really. Let's take a look now and bring ourselves back a little bit to sizes. Let's start with a tobacco plant. Here's a leaf. On the surface of the leaf, we find cells, familiar hair cells. And suppose we were to take a hair cell from such a leaf, and if the leaf were infected, we would find probably within the hair cell a little crystal. And this crystal is a crystal of virus. Now suppose we have a sort of model here of what the virus rods look like when they are arrayed in this crystal. And we see that the virus particles are arrayed in precise order, whether you look at them this way or whether you look at them this way. Now let's suppose we were to dissolve this crystal in water. The virus particles would come apart separately, and we could examine them one at a time. This is now magnified some 250,000 times compared to the virus itself. But a little piece of this virus could be broken, and then if we look at it and imagine magnifying it another 250 times, we then have a particle of the size and of the detail of structure that we see in the model. Now, the protein subunits in the model are seen like this. We can take them apart, but these are really only models. The actual protein subunits have no shells around them. Actually, the chemist knows that he can take one of these shells apart, and that by chemical means, he can actually succeed in finding that that protein subunit consists of a long coiled strand, which is the entire protein molecule making up the subunit. And by chemical means, he can give it a real stretch and stretch the thing out perfectly straight. Well, this is just a model, and we might go to another model, which would give us really more information. And now we go to a pretty large magnification. This model consists now of a series of building blocks that demonstrate what is the detailed structure in this long coil, which is in turn the protein subunit of the virus. And these blocks are held together by magnets. In nature, they are held together by chemical bonds. The building blocks, then, of the long chain of protein, which was the protein subunit of tobacco mosaic virus, are amino acids. And there are 20 of them altogether, shown as numbers going from 1 to 20. Now, in the entire protein of tobacco mosaic virus, there are 157 of these. But it is possible to analyze the sequence of these. By this I mean what number follows what number along the entire chain. And this is done actually by taking the material apart chemically, and then we can analyze these small pieces quite well, and then put together the whole jigsaw puzzle to come out with the whole 157 of the amino acids. Now, how about the nucleic acid? So much for the structural sequence of the amino acids in the protein. You recall from the electron micrograph that the nucleic acid of tobacco mosaic virus is a long strand. And so in the model, it is also a long strand, and it's also made up of building blocks. Actually, the length of the strand of nucleic acid in tobacco mosaic virus is so great that it's about 100 times as long as this little section here. The nucleic acid from tobacco mosaic virus has just 4 different kinds of building blocks. They're shown here by letters, but of course the individual building blocks are actually small organic molecules. In the nucleic acid from some of the other viruses, the letters would be somewhat different. One would be different. In the place of the U, we would have a T. And also the strand, instead of being single, would be double. But the principle of it is very much the same. Now, we have very good reason to believe that if we have a single kind of virus, let's say tobacco mosaic virus, that if we could examine the nucleic acid strand in great detail, and if we could examine the sequence of amino acids in the protein in great detail, we would find that there is complete similarity and, in fact, identity from one nucleic acid strand to another, and from one protein amino acid sequence to another. In other words, for one type of virus, the sequence in great detail of both the nucleic acid and of the protein is identical. But now how is it with viruses of another kind? In other words, let's consider poliovirus. We don't really know the answer, but we have this sort of guess, that if we could examine, and we will eventually, the protein structure of poliovirus, we would find that there would probably be 20 building blocks in the protein structure, but the sequence in which these building blocks occur would be different than with tobacco mosaic virus. And similarly, with the nucleic acid, we would find the same 4, A, C, G, and U, such as we have here, but they would be in different order. And symbolically, actually, it seems to me I could make one virus into another. What I mean essentially is that symbolically, at any rate, the difference between, let us say, tobacco mosaic virus and poliovirus with respect to the nucleic acid might be just as simple as that, taking out 2 building blocks from the nucleic acid from one place and putting them into another place. And similarly, with the amino acids in the protein chain, we might find that this one ought to go here, and this one ought to go here. We don't really know, but still we believe that that is really probably just about the only difference between these kinds of viruses. So now we, in a sense, are ready to come back to the cell. We've gone a long ways from the cell down to these models in which we now have magnifications of about 200 million times. The question that's really important to us is what happens inside the cell when the stuff of the virus enters the cell? And we'd like to know, for example, what gets made first by the cell. Is it the nucleic acid or is it the protein? Now, we don't really know in detail what the answer is, but it seems very likely that the answer is that the nucleic acid is made first. Why? Well, we know now in the case of several viruses, particularly for tobacco mosaic virus, where it was first discovered, that the nucleic acid alone is sufficient for infection. The entire virus does not have to enter the cell in order to cause the plant cell to make new viruses. Only the nucleic acid will do. So if the nucleic acid by itself is enough, then it's likely to suspect that the nucleic acid, upon entering the cell, carries with it a sort of message. It is a message in a code. It's a four-letter code in the case of tobacco mosaic virus, which contains 4 different kinds of building blocks in its nucleic acid. Now somehow, we don't know how, the information that is in this 4-letter code is transmitted to the cell. Whether it is transmitted to the genetic apparatus of the cell or not, we don't know. But after all, the cell itself, with respect to its heritable characteristics, in other words, with respect to its genetics, with respect to the information the body of the cell gets as to what sort of stuff should be made by the cell, the seat of this activity, of this guidance, we believe, is in the nucleic acid of the cell. So it's entirely likely that the nucleic acid of the virus, upon entering the cell, conveys information directly, very likely by just a physical apposition of the nucleic acid of the virus with the nucleic acid of the cell, conveys information to the nucleic acid of the cell as to what the cell should make next. So we think that probably the nucleic acid of the new viruses is made first, but probably not much afterward, the protein has to be made. Now, how is it that we find that the protein of the viruses is in terms of building blocks? In other words, tobacco mosaic virus protein has actually altogether somewhere between 2,000 and 3,000 subunits. We believe that the number is actually about very close to 2,100. Now, why isn't all the protein of the virus all made at once? Why is it that it is made in subunits? We believe the answer to this is essentially an engineering answer. The nucleic acid, which in the case of tobacco mosaic virus contains 6,000 subunits, just doesn't convey enough information in it to allow the cell to make all at once all of the amino acids which have to be made to make all of the protein of the virus. After all, there are 2,100 times 157, which is about 300,000 total amino acids in tobacco mosaic virus, and the 6,000 subunits or building blocks of the nucleic acid just aren't enough to code or give the information for making that many amino acids all at once. So it's like an engineer building a building who will design the bricks of a building, and then the bricks are made over and over again for the entire structure. And so we think that the protein subunits are made individually from the information coded by the nucleic acid, and then these are made in replicate, and finally we come to the assembly. And the assembly process, we believe, consists of just having the nucleic acid get coiled up somehow or other, or in the case of tobacco mosaic virus, arranged in the form of a helix, and then round and round go the protein subunits, assembling the whole structure with the nucleic acid somewhat toward the center. In the case of the spherical viruses, the protein subunits arrange themselves with this marvelous and efficient geometrical nicety that we have seen in the case of poliovirus. We have seen how, in the case of tobacco mosaic virus, we can start with the plant, we can extract the virus particles from the plant, then we can go on to break the virus particles up into somewhat smaller pieces, the protein appearing in the electron micrograph like a donut, which represents a slice across our model containing 20 or 30 of the protein subunits of the model. We have seen how the chemist can break the protein subunits into the individual ones, and then how even these can be pulled apart and analyzed for their entire sequence. We have seen that with the nucleic acid, we do know the constituent building blocks, although at the present time we don't understand their sequence. But we have an approach now to the language of life. That is, we have a strong feeling that it is the arrangement of the building blocks of the nucleic acid that determines the sequence or order in which the amino acids of the protein will be made and assembled. So we are beginning to learn something about life processes that we didn't know before by observing the detailed characteristics of the viruses that are made by cells when the cells are infected with the nucleic acid of viruses. And so we might speak of the long linear array of nucleic acid and the linear array of the protein chain as essentially being the language of life, to be sure for us now in a somewhat coded message. Now, I really haven't been saying all of my remarks in a way that are correct in great detail. There are probably some mistakes in these details, although we believe that our general picture is right. But there are some false places, I'm sure, and there are tremendous gaps in our knowledge. The exciting things for people like me, virologists, are to correct the mistakes that we have in detail and to remedy the gaps in our knowledge.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. Robley C. Williams, Professor of Virology. The series was made possible by a grant from the National Science Foundation. This is National Educational Television.